Hi class, welcome to our virtual activity on DNA isolation using the solid phase extraction method. First, we need to prepare your sample in a sterile microcentrifuge tube and label both the body and the cap. So in this activity, we will be extracting again DNA from a bacterial broth culture. So we take 1 ml of broth and dispense it into the labeled tube. We then now centrifuge the tube to create a bacterial pellet. Now, after separating the bacterial pellet and the supernatant, decant the supernatant into the waste container. Then, we thoroughly mix it using the vortex mixer. Next is to apply the lysozyme. It is an enzyme that aids in the lysing of the cell. So we add 10 UL of the enzyme and mix it again using the vortex mixer. After mixing, we then incubate the tube at 37 degrees Celsius for 20 minutes. After incubation, we then take out the tubes and centrifuge it to separate the pellet and the supernatant. Always balance your centrifuge before you use it. Next is to decant the supernatant. We then resuspend the pellet using the buffer R2, which is also provided by the commercial kits. 180 UL of the reagent or the buffer will be added to your pellet. We then add another enzyme, which is proteinase K, an enzyme that helps in digest proteins. 20 UL of these enzymes will be added to the resuspended pellets. Then mix it using the vortex mixer. We then incubate again the tube, but this time at 65 degrees Celsius for 20 minutes. You may shake the tube occasionally every 5 minutes. This will help expedite the enzyme treatment. At the end of the incubation, the solution should be clear or extend the incubation time. After the enzyme treatment, we will add another buffer, which is buffer BG. This will help homogenize the solution, or in this step we can now call as lysate. A lysate is a solution that contains lysed cell and its products. 400 UL of the buffer will be added to our lysate. Mix thoroughly using the vortex mixer, and then after that, we then incubate it again at 65 degrees Celsius for 10 minutes. After the incubation, we then add 200 UL of absolute ethanol to our lysate. Gently dispense the absolute ethanol to your lysate and mix it immediately through pipetting. We then now prepare our assembled spin column and collection tube. Label it appropriately. Once that is done, you can now transfer your lysate to the spin column. Gently dispense the lysate onto the spin column. Do not touch the tip onto the sides of the spin column, nor poke the silicon membrane at the bottom. Then centrifuge the tube for 1 minute. And again, always balance the centrifuge first before you start running it. Now, after 1 minute, take out the tubes out from the centrifuge and notice that there is fluid inside the collection tube. That fluid is now called your flow through. We will now discard the flow through. In discarding it, gently detach the spin column out from the collection tube. Make sure that you do not touch the silicon membrane. After discarding the flow through, you gently reinsert again the spin column onto the collection tube. We then proceed with washing. We use a wash buffer, which is provided by the extraction kit. We add 650 UL of the buffer onto the reassembled spin column. We will centrifuge again the tube for 1 minute. Make sure that the centrifuge is balanced before you start running it. After 1 minute, gently take out the tubes. Discard the flow through. Be gentle when you detach again the spin column and discard the flow through. And when you reinsert it again, make sure that the silica membrane of the spin column does not touch anything. Centrifuge again the reassembled tube, but this time without the buffer. This is a crucial step to dry out the membrane, so to avoid carrying over any traces of wash buffer. And again, make sure that the centrifuge is balanced before you start running it. After 1 minute, gently take out the tubes from the centrifuge. And here, you may notice that there is a small volume of flow through inside the collection tube. These are traces of buffer from the membrane that we avoid in carrying over to the next step. Next, prepare a new sterile microcentrifuge tube and label it appropriately. After labeling the microcentrifuge tube, gently take out the spin column from the collection tube and insert it to the newly labeled microcentrifuge tube. After that, we can now proceed with DNA elution. We add 100 UL of the elution buffer to the spin column. It is best to dispense the elution buffer directly onto the membrane to maximize the elution of any bound DNA. We centrifuge the tube again for 1 minute. And always make sure that you balance the centrifuge before you start running. Now, after 1 minute, you can take out the tubes and notice that there is flow through onto your microcentrifuge tube. Your extracted DNA is inside that flow through. After the complete centrifugation, we can now discard the spin column and keep the flow through inside the microcentrifuge tube and store it appropriately. That ends our activity for today. Thank you for watching.